especially linking individual behaviors in brain structure and function and behavior is what defines Birte's work. And when we at the IAREP decided that, and at the local organization decided that this would be an excellent way of starting the conference, I explicitly talked to Birte and we requested to emphasize the relation to decision making because we are all decision scientists here. And this is really the meeting point of economics and psychology, but a lot of us also have at least part of a foot in neuroscience. We would like to become more of a meeting of economics, psychology, and neuroscience. And I think that for this purpose, the talk of Birte is going to be perfect. Birte, it's a pleasure having you here. Thank you very much for being here. The floor is yours. Take the time you need.
Great. Yeah, before I start, I'd like to issue what an honor it is to be here and to join this conference, the 50th anniversary. It's my first time at IAREP. I was really honored to receive this invitation by Carlos, and I, of course, also would like to thank Jaume and everyone who organized this meeting. It's my second time in Spain. I don't know why, because looking around, it's incredibly beautiful, so it won't be the last. And yeah, I'm very happy. I've been only here already for 20 minutes, and I feel at home, to be honest. I have had the chance to talk to some of you and to inquire a bit what everyone is doing, and yeah, it's really a great pleasure. And as Carlos just mentioned, so my background is deeply rooted actually in the cognitive neurosciences. That's where I started out. In fact, in Leipzig. I'm originally from Germany, and I did my PhD at the Max Planck Institute at Leipzig, which is really basically my scientific cradle, if you want. Where I was supervised by these lovely men, Marcel, Yvonne Cramon, and Irin Koch, who taught me basically the nuts and bolts how to conduct cognitive neuroscience experiments. And then about 20 years ago, I moved on to the University of Amsterdam, and I'm still located there. And I had the pleasure to start working with Richard Ridderinghoff. He's more cognitive control researcher, and Eric Jan Wagenmakers, who maybe some of you actually in the audience this name recognize, because he is not in economic psychology, but in mathematical psychology, which is a rather small community. But it was a true eye-opener for me to start collaborating with people from that community. And in fact, it really elicited the interest in moving not away from brain research, but basically trying to understand and starting to ask very different questions than I was used to ask. And that is not necessarily where in the brain, by means of blobology, things are happening, but actually trying to understand more mechanisms in the brain. So how does this actually work? And during the years, in particular EJ and I, really early years, we started to develop this model-based cognitive neuroscience framework. And that is at the core of the research that I'm presenting to you today, which is primarily focused on decision-making processes. So everything that I'm kind of talking about is really focused on decision-making networks, substrates in the brain. But we're not just simply using an fMRI machine and then do some experimentation. What we started to think about, and again, it is a highly interdisciplinary approach by using quantitative mathematical models. And in particular, I'm using evidence accumulation models. For example, the diff diffusion model or decision diffusion model, which I'm sure many of you have heard of or even maybe worked with. And these are very powerful models that allowed me to actually really zoom into the latent level of decision-making on an individual level or even trial-by-trial level. And so the idea really is that we're not just conducting behavioral experiments and infer some cognitive processes such as decision-making, but what we wanted to do is we wanted to fit formal models, mathematical models, to behavioral decision-making data, so to entire reaction time distributions. And this allowed me to estimate latent variables for speeded decision-making and other biased decision-making, to then use these parameters in a unidirectional way, first of all, with my neuroimaging data. And in fact, this is a placeholder. You can really use all sorts of modalities. We have used this with functional MRI, with structural MRI, with diffusion. You can use it with pupillometry. You can use it with really exciting, more peripheral measures of EMG or MEG. Basically, the entire range of neuroscientific methods that you can think of. And the power really lies basically in these formal models, not necessarily in this imaging part. But still coming back a bit to my background, and that has inspired really a lot of people. So we're calling this basically an integrated model-based approach by now, because these people, most of them have different backgrounds. They are psychologists, they are cognitive neuroscientists, they're engineers, they're mathematicians. So a full range also of disciplines that basically joined our efforts of extending this approach, extending modeling, extending the imaging. But so I'd like to take you on a little detour for a moment, and I'd like to take you basically on a journey that I engaged in 10 years ago when I realized that brain sciences really has a huge problem. I'm going to explain what that exactly is, and this problem is not solved. But I hope also to provide some solutions that may remedy the situation. The second part is more to present you with this theoretical framework, how we can bring together brain imaging or brain data. And again, this is, you can substitute this with fMRI, SRI, whatever you want, and behavioral correlates of decision-making in my case. And at last, I'd like to present to you a concrete example. So how does this exactly work, and how can we do this in an empirical setting? But so to the problem and to the big surprise, and it was really an eye-opener, a rainy day in Amsterdam, as it mostly is, reading and looking through Neuroimage back then, and it was a Neuroimage paper published by Heidi Johansen-Berg. She's a fantastic neuroscientist from the University of Oxford, kind of a light figure in my growing up. And she wrote in this paper, it was a quote really, that she was herself surprised that most imaging studies, functional, structural, you name it, have been focused on the cortex. And I'm sure everyone knows cortex, the mental, everything, like very important language, reasoning, all of that. But there seems to be not an accident in that sense, and it's also not intentional, but there seemed to be a neglect in that most of the structures below the cortex, so in the deep brain, and also in the brainstem, this is the brainstem and this is the cerebellum, they simply have been neglected. They are simply not reported. So it seemed that we are looking at decades of corticocentric research, also for decision-making, not only for higher cognitive functions. And so I was really astonished when I read this and thought, okay, what is happening? Why is she saying that? And because also during that period of time, there were very seminal papers coming out by people from the group of Van Essen. He's a very prominent figure in the US, a neuroscientist figure. He said, well, we know exactly the number, how many cortical fields we have. It's 180. And I thought, wow, that's clear. You usually never really get such concrete answers, at least not really in the neurosciences. But this is what he came up with beautiful technology methods. And this was corroborated also by anatomy. And I thought, okay, so Heidi says this, they say they have the answer, so how many are there actually? And then I just counted. So I went to the Holy Bible of Anatomy, which is this, and I just simply counted. And so I came up to a striking number of 455. And note that these regions, we know them. I couldn't have counted them if we wouldn't know them, right? So they are anatomically defined. But then I started to really get excited and went to, let's say, classical brain imaging analysis tools. We have standalone softwares that everyone basically in the field uses, called FSL, SPM. So I just simply went to them and I inquired, so how many are actually incorporated? If I were to do, for example, economic decision-making task, I'm interested in investigating what's going on in the brain, and I'd like to also understand what's going on in the deep brain. How many can I actually inquire about? And then the striking number of 7% came up. And so In a nutshell, what it means is that we're looking at a problem that I call terra incognita, if you want, so uncharted territory, meaning that basically all areas here in gray of the world map are uncharted. We don't have atlas maps. We do measure them, right? If I put you in the machine, I get signal, but basically I don't exactly know where it's coming from, and there are many other problems I briefly discuss. But so these 7%, we are talking a couple of years later now, maybe we're 10%. This is what we have access to. And I paint now Europe, and Europe, I thought that's not, you can put this, there's no political statement here. It's just kind of a visualization of what's going on. And so I wondered, oh my God, what have we done? What are we doing? I'm doing brain science. I thought I'm measuring the brain, right? And I have information about it, but apparently that is not really truly the case. And in fact, it is not necessarily the case. And what is also striking is a theoretical exercise I did with one of my students. It's a quantitative meta-analysis. And I wanted to understand whether this observation has already translated actually in the neuroimaging literature, and this is exactly what we found. So what we did is we harvested PubMed, and we harvested over tens of thousands of papers by means of mesh terms, looking into linkages between cognitive processes, for example, decision-making. There are also decision-making processes that many of you study here, and brain structures. Meaning in the deep brain. So I took all these 455 structures and I inquired whether they are linked and reported in the literature in these tens of thousands of papers with any of these structures. And what we found is, intuitively, I think you immediately understand it, but I'm very worried, is that basically the probability with which a cognitive topic is mentioned in association with a particular structure is clearly biased towards the size of the structure. So just to kind of do a little short inquiry to understand, how many of you have heard about, for example, the thalamus? Excellent. So I don't want to bore you with brain stuff, but it's kind of a little detour into also understanding really the problem in brain behavior linkages. So basically what happens here is the thalamus is one of the biggest structures that you find down there. Now you can parcellate the thalamus. There's huge debate in the neuroscience house. Some people think there are 80 substructures, others think there are 6. So it's a huge debate, totally not clear. Anyways, if you just take the thalamus as a whole, it's huge. Everyone can see that. I show you a picture, and you have never seen a brain picture, you know where it is approximately. But then the striatum also next to that, again, a very prominent structure mentioned in the literature. Simply, I argue, because of size. Yeah, so people are pretty sure that this is in that region. And then, if you are a bit more interested in anatomy, you see that this becomes more and more complicated. So the argument again here is when you start to investigate, you'd like to set up a study and you hypothesize about certain regions, be aware that there is this bias in the literature of linking basically to large structures because they simply could visualize that. And so, obviously, then the next question is, why is this? Why did this happen? Again, it was not an intentional gap saying we love cortex. Cortex is so important and it differentiates it from other species. It certainly made us in some aspects. I don't argue about that at all. I was more interested in, from an imaging perspective, why such a large knowledge gap? And again, quite intuitively, when you look, for example, at this section, it's basically, I just basically display this plane cut approximately at this level, and then I zoom in the deep, I think it's axial, like this, and I just zoom in on that level. And then I get to the deep brain structures, some of them. The abbreviations don't really matter. We're looking at certainly nuclei that are super important for decision making, we know that, and also economic decision making. The problem here is that they are incredibly small. So the thalamus is big, you can make it smaller and smaller, but some structures, for example, the subthalamic nucleus, some I've heard of it, is just in itself not bigger than the nail of my pinky. Yeah, and then you also see these weird shapes. Yeah, this is not a nice patch in the brain, but these are, let's say, very individual nuclei. The next thing is that, as you can see, they lie in incredibly close proximity, and that is really throughout the entire subcortex. It is, when you look at the slices, not clear there's a border. You can't just look at it and then say, of course, this is Switzerland and then this is Germany. It really depends on understanding a bit more about neuroanatomy without going into detail to discern the stuff. And the last thing, just as a short mention, and I didn't know that, and with that non-knowledge, I actually delayed the project of one of my PhD students substantially, because I told him, the data is great. We put a lot of effort in it, the results, this must work. And he said, Birte, this doesn't work. It does not work. There is no signal. You're measuring noise. And so we went back and forth for half a year by me telling him, we measured it to the best of my understanding, until I realized that all of us, we have heavy metals in our brains. For example, iron, and we also carry neuromelanin and copper and lots of stuff. And I'm mentioning this because it interacts how you can visualize in the first place structures in the brain. Now that goes very detailed, but it has a huge influence, and I think pictures often tell much more than words. Here's a concrete example of what I mean with that. So these are just brain images from healthy people, and these are just different MR sequences. Doesn't matter what exactly it is, but this stuff and this stuff focuses more on iron content in the brain. This doesn't. Now, if you, for example, just for a moment focus on these round blobs here, this is the red nucleus. It is clearly visible, right, in this contrast, MR contrast it's called. If you compare this to this one, it's gone, right? You don't see a thing. And this is the contrast that most imaging studies report. Yeah, T1 weighted it's called. This is the classical vanilla contrast. When you open imaging neuroscience article these days, or former neuroimage, you will see this is reported, this and this not. So as if people maybe not know, and it's standard. So I'm also not saying it's wrong, but I'm just saying if you're really interested in brain and also these structures that give rise to decision making, then yeah, this is very important to keep in mind, just as an example. So we looked at all of this, and it gave me a very deeper understanding how complicated really neuroscience also is, of course, and cognitive neuroscience, and to do it maybe a bit better. And also, in fact, again, to remedy the situation. And what we started to do is to create an automatic segmentation algorithm without going into a lot of depth. We're only at 63 structures of 455 at this point. It's pretty bad, I know. And I calculated how long would it take us with this approach to get to the 455, and even submitted a paper, a pre-submission inquiry at some point to a journal, saying, look, this is what we did. This was the time investment. And they came back and said, yeah, sorry, we want all of it. Why are you going half this way, or not even half? And then I said, well, I calculated it would take at least 3 lifetimes, and not only from one person, to basically get to that point. Of course, you can argue, well, Birte, this is really kind of a good way forward. Maybe it really is not, but I can assure you that it's the most detailed try to remedy it. And also this algorithm, noteworthy, works on really all types of data, on 1.5 Tesla MRI, on 3 Tesla, on 7 Tesla. You don't need special machinery. You don't need special contrasts. It is really the possibility of opening up the field to investigate these deep structures and to inquire about their functionality. But it is a long way, obviously, to go. We started with structures that we knew are important for decision making, including the basal ganglia and some other structures. It's a start. But then I end with my brain pictures and my little detour coming back to behavior and decision making. I just, if I start something, I just get excited and then maybe also a bit derailed. But yeah, I realized with in vivo human data and the resolutions that I can acquire these days, I'm working with ultra high field MRI. So these are one of the most strongest MRI fields that you can have. There are about 150 machines in the world. they don't deliver what I need for these smaller structures. So then I started collaborating with an anatomist, and she's fantastic. We developed a pipeline. I'm just briefly showcasing this, where we collect post-mortem brains from people that are otherwise, let's say, they were healthy. They are deceased, but they are not. They are brain healthy. So eventually, their demise was due to a heart attack and other reasons, except for the brain. And we realized we need this detailed information because we want to submit them then to an extensive scanning protocol, which we did, for weekends. So this wealth of data of 6 brains by now, or more, I'm not sure, that were submitted to an extensive MRI scanning session. And then we sectioned these brains in 200 micron sections. So 200, just imagine how small this is. It's still, for brain anatomy, it's big, but for this whole brain approach, very small. And we started to basically cut them up in order to look really into cells, fibers, to learn on the microstructure what's happening. Again, also for the delineation. But most importantly, again, all this research is not meant as monodisciplinary now, learning about brain anatomy or anything. I want to go back to the function. I want to learn about decision making. And the way to do that is that we reconstructed these histological sections into 3D again. Yeah. So basically, we photographed each section, and then this 3D reconstructions were computed. And with this last step, it is possible to use all of this detailed information, microstructural information, again in your in vivo situation. You can simply take this post-mortem brain, you can put it on top of your living human brain, and what you then get is an incredibly highly resolved entire human brain representation for different cell bodies, for fibers. They're all combined in the same scale. And I now can learn on a much higher resolution where is what, and I can delineate eventually in my own subjects that are undergoing my experimentation, actually also new structures that we haven't mapped yet and that you maybe find interesting for certain problems that you think are relevant. And I should note that everything that I'm presenting to you today is freely available. You don't even have to leave your email address, anything. I just warn you that this type of data is very large, so you'd better make sure you have enough storage space in order to get it down if you're ever interested in working between the translation of this level of detail all the way into the human situation. I'm briefly skipping it. This is more about the physics and the iron and how to measure it. But as I mentioned, when you're interested in deep brain structures, it's very important to optimize and not just take off-the-shelf sequences that are implemented at scanner sites. So I stop here for a moment because that was my little detour into, let's say, the realm of brain and brain anatomy, and just showcasing how much we tried to remedy the situation as to get a better traction on the whole brain and not just cortical representations and larger fields. The original idea I started out with, and this is still at the core of my interest, is to understand how decision mechanisms are implemented in cortical, subcortical networks. And there are some proposals, for example, by Yassine Temel and others, that differentiate between 3 different networks. So one motivational network relevant for decision making, a cognitive and a motor network. What we see is that there are not only different cortical representations eventually implementing this behavior, but you see, without naming now all these structures, that they translate information by means of excitatory, inhibitory connections into the deep brain. So we see that there are very distinct networks that eventually give rise to different types of decision making. And I've looked at your program, and I'm sure if I would sit down and someone would explain to me what the specific task is or interest, that one could make very specific hypotheses which aspects of these networks could potentially be involved in the type of decision making you're investigating. But I am mostly interested generally, basically, in very basic decision problems for now. The concrete example goes a bit beyond that, and there I involve also reward-related decision-making. But for now, bear with me as to also explain for a moment how joint modeling now could work. Because what we wanted to do is we wanted to go beyond simple correlation. I have some brain measure, and I have some behavioral measure, and then I compute a correlation. What I like to do is I'd like to extend this original approach that I showed you, right, where I'd used formal models, and I model my behavioral data, and then I inject it basically in my imaging analysis. My aim, or our aim now, was, in fact, to truly come up with a generative joint model. And eventually, this is a Bayesian implementation. On our side, there are many different ways here to roam, many different also suggestions how to do that. But at the core, it's always the same. We don't just want to collect behavior on the side and then brain on the side, and then somehow afterwards bring this together by correlation analysis. We'd like to, in the end, and I'm not there yet, maybe others are, actually have one model that explains these two modalities. Because when you scan, you also collect that behavior at the same time, right? This comes from the same organism, obviously. So in that sense, it would be beautiful to tightly link it, I think. But so the models that I use and that I also use for this joint modeling framework are predominantly evidence accumulation models or sequential sampling models. I ask a bit around, how many are familiar with these types of models, diffusion modeling, evidence accumulation modeling? Okay, thank you. It's very intuitive, I think, again, and that's also why I liked it with my background. I could understand somewhat why mathematical psychologists are so fond of these models, and they are. They have been around for 50 years by now, so they are very powerful models. They are well parameterized. We're looking at a space of 8 to 10 parameters, for example. And they are extremely powerful in making excellent predictions for behavior. And that means behavioral reaction time distributions for correct and erroneous responses, and have been used in all sorts of different decision-making problems, including biases, including speed, including rewards. Basically, everything I saw in your program, I'm pretty sure that these types of models could eventually be insightful, useful to be fit to such data. What happens in these models is really simple in that sense. The assumption is that, of course, when you work on a problem, on a decision problem, that, of course, first of all, you need to encode the stimulus. You need to encode the problem, right? You need to see what you're actually supposed to work away from. So physiologically speaking, this is the so-called encoding time. It is the time that simply this what you see on the screen or what you see in your environment hits your retina and just is basically processed into visual cortex. It's a peripheral process, potentially. That can be debated, but it is basically just looking at it, if you want. Then at the end, of course, if I ask my people or subjects, please indicate your choice, they need to press something, or they need to make it a card, or maybe use their foot, they need to do something. That time, basically from motor cortex along the corticospinal tract into the effector, is also taking up time. And that time, again, is also in the periphery. It has nothing to do with this decision itself, but it is basically something that I want to get rid of. And if I just look at one point estimate, so for example, one reaction time that I collected in my experiment, I cannot do that, right? It's all smeared, all of the stuff is in this one time. But now using reaction time distributions in these models, what I can do is I can start to disentangle this. So basically, and I know if a mathematical psychologist like Francis Thürlings would be here, maybe he would not be so happy if I say this now, but basically the way I treated these peripheral processes, encoding and motor execution, I basically dumped them in one parameter, the so-called T0 or TER parameter. That is really a point estimate how long these peripheral processes take, and I want to get rid of this. So I basically put them into the trash bin, if you want. Yeah. So I fix them, and I say, you are not part of what I'm estimating here. You are not part of the prediction that I like to form, because what I'm really interested in is this latent decision process. And in this implementation of the sequential sampling models, this is just one way to implement it is. That I implement racers. So I have as many razors. This is one razor for a right response, for example. This is one razor for left response. You can have as many razors if you want, as many choices that you have or implement in your experimentation. This needs further discussion, but for now, I just would like to leave it as such. So basically, what I'm doing now, I would like to understand the decision-making process that happened latently, right, in your head by parameterizing exactly this process. So in a nutshell, the idea is you see the stimulus, and the stimulus triggers the accumulation of information, right? So you have to process what you need to decide on, and I can parameterize this by the means of a drift rate. The drift rate V, which is commonly used denominator in all of these sequential sampling models, is a very prominent parameter because it basically reflects the quality with which you extract information out of your stimulus. So that is one parameter. Another parameter is the so-called response threshold B. The idea is the decision starts at this point. The steepness of the slope reflects the drift rate V, and then eventually it hits a response threshold. And as soon as the response threshold is hit, a response is issued, meaning executed. That's basically, in a nutshell, what's going on in this model. And what I can do now, I can estimate these parameters. I can vary, for example, the drift rate, inquiring whether my subject actually worked through my task more on a perceptual level. Yeah? So this is a totally different process than if I think that my subjects voluntarily change these response thresholds. So if they change these response thresholds and lower them, the prediction is that you become much faster. But the prediction is also that you are much more error prone. So there are very specific predictions linked in terms of behavioral outcome to changing these parameters in the model. So what you learn with that when you generate predicted responses is strategically what did your subject actually do? Yeah, because you can't observe it. Even with my fMRI machine, I don't know what's really going on. I don't know how they solved the problem. And they could have solved it in many different ways that you are not aware that they did that. And it's amazing how individuals vary how they solve these problems. Yeah, you think I have full control, they sit in this experimental room and I know exactly how they should solve it. And then it turns out when you fit the model, you see that they did something completely different than what you expected that they should do. And this is exactly what you wanted them to do. So this is exactly one approach how to tackle this. And as said, these models are extremely powerful for many different experimentations, for many different decision-making problems. And I'll also in the concrete example show you how they beautifully can be extended to learning problems, how you learn outcomes, and how that impacts, in fact, behaviors on a trial-by-trial basis. I'm also really happy to take questions if you like to make this more. Yeah.
Your picture reminds me much more of linear holistic application of I know about all the discussion of whether the engineering side will say, what's your take on that?
Yeah. Thanks a lot for this great question. And sorry for not going into detail, also maybe for the sake of time, but you are absolutely right. This is the racing model. So exactly, it has features of the LBA, the linear ballistic accumulator model. What Carlos is asking is, there are different types of sequential sampling models. I think the drift diffusion model is one of the most prominent ones that you may have heard of because it has been around for the longest also. For example, proposed by Roger Radcliffe. May sound familiar or not, it does not matter. The implementation of the diffusion model, you could argue, it is first of all different. It is biologically, you could argue, more plausible because it does not have racers. It actually starts, you don't have, I would love to paint this, but I'll just, I just show. So basically, instead of having two racers, the diffusion model, what happens is you have two bounds. Let's say this is the right bound, the right response, this is the left bound. And then you have in the middle a starting point. So instead of having two racers, you only have one noisy decision process. And that starts, if you don't have biases, for example, like I don't tell you, hey, response A is much, much better than B, I can shift you around. I can basically shift you to closer to that bound or that bound. So in a diffusion model, such a bias, for example, would be implemented by having the start point. So this point start much higher towards the threshold, and then it accumulates. So we only have one noisy accumulation process. This model has more parameters than a race model, and that was also one reason why we chose for this implementation. It is more parsimonious. But what is also, I think, important to note is that the many different variants, in particular the full-blown, full-fledged DDM, the fusion model, or these racing models, they mostly come to the same predictions. Yeah, we're not talking, these are the same classes of models. And it is really nice when you use these models, of course, also to do model comparison. And eventually you will see that race models are quite powerful given they have fewer parameters, so they will outperform the DDM, simply because they are more parsimonious, still having a very good fit to your data. But yeah, it may, when you start reading about it, may be a bit confusing because there are these various implementations. But I think the most prominent ones are either race models, or the noisy stochastic diffusion model process that I hoped to signal to you here right now. If you'd like to learn more about it, please come, and I can also provide really very nice references. I think that very clearly lay out what each model is doing and for what purpose, or what benefits you get out of it. Yeah, but our reason to use this is basically parsimony and, in fact, a model fit. Yeah. Yeah. Oh.
Signal detection theory, so if you could explain the difference.
Yeah, thank you very much. This is the question is about response thresholds and response bias and how we can potentially differentiate between those. And I think this is really a question also for some sessions that may be of relevance. It is. Yeah, I wish I had a chartboard. It is much easier to explain if I have the diffusion model than these representations. But in a nutshell, I think I may explain this by means of experimental manipulation, first of all. So these response thresholds are, first of all, to be believed under voluntary control. I think that's very exciting because people can voluntarily, intentionally adjust those. If I tell you, for example, globally, independent of condition, I would want you to perform super fast. The idea, the hypothesis concretely for these models is that these response thresholds move down globally, independent of condition. Yeah, so this is a global reduction of basically getting your system in readiness mode. Response bias, on the other hand, is very selective. The idea here, and that's not captured in this response threshold parameter. I have another parameter actually for response bias, which is really exciting, I think, and easier to represent, not in these racers, but in the fusion model. So basically what happens is consider you have these response thresholds. So now not separately, but let's say here's one threshold for one response, here's one threshold for the other. And so your experiment starts, right? And I don't give you any biasing information at the beginning. Yeah, you don't know. You just know that you have to. There's a stimulus, and you have to decide something. The assumption is there is no bias, so the process, the decision process, starts in the middle, right, of the two response thresholds. But now response bias comes into play. If I tell you this response is much, much better, you get much more money, right? then when you respond this one. So the assumption, the hypothesis then, instead of doing something with this response threshold, it stays fixed, I move the starting point up. So that's a different parameter. So you can disentangle basically this shift at the beginning of the decision process, and you get an estimate how much bias was there, right, compared to looking at the fixed response thresholds. And so then you can differentiate between a global, let's say, response speed regime, and you can selectively test how much prior information your subjects took into account to perform much quicker selectively on one dimension compared to the other. And that is also, I think, the beauty of these models, because again, if you just look at the one reaction time point, you will not be able to disentangle any of this, right? You just see, oh, the subject was quicker. Sure, you can condition then on reward or something, but this model allows you to clearly differentiate whether people just got in this excited speed mode to collect stuff, or whether they actually really paid attention to the bias that they were presented at the beginning. Pretty much like, I really tell you, look, I give you 2 buckets, and I tell you the right bucket has more incentive than the left one. Surely you will be favoring the right one if you trust me. Well, there's another, you study this, I never studied trust. I don't know what, this is complicated, I think. Anyways, let's say you trust me, I'm trustworthy, and I tell you this. Then the idea is I can disentangle, you shift not only your attention to this, you actually shift your entire response regime towards the preferred option. And then, of course, you could ask many more beautiful questions. You could manipulate trust and many other social dimensions and thereof. But you know for sure that it is not simply that your subjects speed up or do all sorts of strategies, really, that you have no control over. Does this answer a bit the question? Thanks. Great. I'm really happy to yeah. Yeah.
Thank you, Birte. This is a very interesting talk. Then I'm an economist, and you say that we can, many problems that we study are also relevant for this. Then in your task, or here on the slide at least, there's clear right or wrong response in a sense. But then, of course, when some decision problems that we study are more like compensatory utility-based, so you make trade-offs or not, so you don't know per se on a subject level what is best or the worst option. So how can you extend that to these kind of settings?
Yeah. Awesome question, and I hope I can answer it. My concrete example is exactly that, and it's if you want, well, new is always relative, but maybe I just go there. Yeah, I just go there because I'll explain this later. That's exactly what we wanted to do, and that is exactly what lives in here. Surprisingly enough, these decision problems that I started to study, right, they are arbitrary. I mean, monkeys can do them, that's nice, mice, and perceptual discrimination is important. I mean, we live in a perceptual world, so yeah, sure, it was a good start. But then thinking about it exactly, that makes no sense. Behavior is not based on just navigating your world left, right, right? Of course, you learn, and you learn in your context. So there are dependencies, and that brought us actually to the problem of reinforcement learning, which I think is related, without making any assumptions of economic problems, because I'm in new waters here, but I think most of you know reinforcement learning paradigms and of course modeling these problems. And the simple idea was to overcome this idea that the trials that I model with IAREP, so evidence accumulation models, I assume they're independent. Yeah, that is the core assumptions of these models, and there is no learning. So I'm looking at basically distributions, and I don't take into account any dependencies. In reinforcement learning, obviously, that is not possible. This is the core of reinforcement learning, that you learn based on your choices, right? And that you maybe learn also based on probabilistic feedback and many different manipulations. So that what it means basically is to marry these two research areas, really, because reinforcement learning up to a couple of years ago and sequential sampling models, they were living in their complete isolated silos for decades, right? I think they like each other. I mean, I've never been to a conference where I could even imagine it would be great to have a conference to bring them together. And in fact, maybe it's this one, because it took us quite some while, and not me, in fact. This work is really done by Andrew Heathcote, for example, mathematical psychologist, and David Miletich, who thought for a long time, how can we accomplish this mathematically also, yeah? How can you basically inform Q values, learning parameters with these diffusion perceptual parameters, yeah? Because that also plays a role. And when you only look at reinforcement learning and probabilistic learning, there's no account for how exactly that is actually accomplished in terms of decision making. Yeah, so the quality of decision making. So we thought this has great merits if maybe we create a model that combines these two realms. And this is just one proposal. There are a couple of more papers. It's I haven't seen this to really catch a lot of interest, which is surprising because I think it has such a great potential to generalize from the specific perceptual discrimination and economic or reward-related problems. But having said that, so the way it looks like, so first of all, maybe I jump to the task, and maybe you recognize a bit of the task that you guys are working with. But so, yeah, we basically really married a manipulation lent from perceptual discrimination. We know we can very well model with not SEMs, but IAMs, so evidence accumulation models, and a manipulation that has been shown many, many times to be well modeled with reinforcement learning. And so what happens in this paradigm is that we had a short fixation, and then we presented our subjects with either a speed cue or an accuracy cue. A classical speed accuracy manipulation. I tell you be fast, I'll tell you be accurate. And that is important because often you have more qualitative choices, right, in these economic problems. There is no speed, and you do need that speed. You need the latency of responses in order to combine these two worlds. Other than that, of course, you can do many other things. You can do D prime. You can do many other things. But the idea was actually to go to the full reaction time distributions. So then the speed and accuracy cues were followed short fixation, and then they were followed by these arbitrary stimuli. And subjects had to choose. This is an arbitrary choice. Now you can imagine you show a car, or you show a house, or you show different economic problems. This is absolutely flexible. Important thing is that each of these stimuli is associated with different reward structure, and people have to learn it. So it's not only that they don't know, right, and there are different probabilities in that sense, but they also have to learn over the course of time what is preferred. And that, of course, we hypothesize has an influence on the speed of their choices. And, of course, eventually also makes a very concrete prediction for error rates. Now, all of a sudden, you can actually really quantify how, for example, the speed regime or like this in these settings have an influence on your economic choice. Yeah, you can even maybe push them around. You can manipulate them in that sense. You can bias them, and you can, by using this joint model, clearly indicate where it's happening what. And mathematically speaking, this is how it's spelled out. So this is really the integration of, again, we're working in the race model realm here. So two racers, two choices, left, right, if you want. And then we're estimating the decision for the first racer and the second racer. And then what we're looking at is where we're hypothesizing that in this particular experiment, in this particular setup, that the response thresholds, which are affected by speak accuracy, we know that they are selectively impacting on the learning rates. Yeah, they have an influence on how quick you actually learn the association between a particular stimulus and the outcome, the reward. And of course, they also get feedback for their speed of the responses. This is just one way and one approach how to do it. You could, for example, also ask questions. To what extent the quality of the information you provide in your economic task, right, it has an influence on learning and on the decision quality itself. Yeah, so you really take into account the context on which your decision is based, the quality of what's seen, and the influence on how that decision then is informed and how uncertainty is also taken into account. That's kind of the idea. Yeah, but it's as said, it's early days. But I do see really merit in it to extend it to more actually ecologically valid problems, because I think this is what you are very interested in, and me too. But as I said, I started out very basic with these tasks. But there is, I think, truly potential in playing around with these types of models. Let's see whether I can still. Yeah, I go briefly back because I think I need to explain for a moment just this graph, because this is the way we're representing our joint modeling results, and that is not the usual way I think that you're used to, if at all, looking at the joint representation of behavior and brain imaging data. It is rather abstract. But just very briefly, it's basically a covariance matrix, and this is relying on the joint modeling of behavior and brain. And so simply this is a brain-brain correlation, and this is the behavior-behavior parameter correlation, this is the brain-brain correlation. Speaking that we look into signal from each of these different regions of interest, and then of course they're correlated against each other, so that's that. And we also were interested, this is going back, I have to go back shortly for this graph. This is going back to the discussion of response thresholds and parameters, because the most common implementation, as I just explained a little bit, I tried to explain, is that you have fixed response thresholds. You have a bias parameter and you have this drift rate. But then 10 years ago, approximately, there was a super exciting discussion coming up that these fixed response thresholds make no sense. And that was led by, for example, Chiani and Shetland, working with non-human primates. What they argued is, this is all wrong. 50 years of fitting is wrong, or 40 years. We shouldn't assume that there are fixed thresholds somewhere implemented, that this is the right fit, it makes no sense. What we need is a different implementation of these response thresholds. So instead of assuming that there's this noisy process or this ballistic process shooting up to the threshold, you execute a response, right? This is collapsing. So it's not a horizontal line, but basically what we're assuming is these bounds, they just come in. And what that predicts is that you start to discount information. So the longer you wait during one decision trial, you basically start to just discount what you think is coming up later. And you think, okay, I had it now, I want to get out of this experiment, I just press a button, I make a choice. I don't care whether I think it's correct, let's just go. And that can be, this prediction can be accommodated by assuming that you're not fixing it, but you're actually just letting it collapse in this way. Mathematically speaking, this is not totally non-trivial, because you can also assume, instead of letting this collapse, you can put a gain factor on the drift rate. So you have a fixed bound, you just have an additive factor on the drift rate, it does basically, mathematically speaking, the same. So it makes the same prediction in terms of reaction time. May it be as it is, and I cannot I'm not interested. I'm not a mathematician. I cannot discern this. I cannot dissociate it. And I didn't want to, but I was really excited or interested in why do they say this after 40 years of a very well-established model. Right, let's put this to the test. And so in one study, we analyzed data from this lab, from the non-human primates, and the results looked exactly the same. The point was not that they maybe used the model in an incorrect way or anything, but we compared it to human behavior. And so what we found is that humans preferably actually have a fixed bound, while animals show this collapse. So in fact, it is true that in this type of experimentation, you see that non-human primates prefer a collapsing bound model compared to humans in many experimentations where we see the classical implementation. I'm telling you all of that because this is really important in terms of mechanisms. Again, how is this choice made? All right, is this driven by this collapse, or is it actually parameterized by a fixed bound? And one speculation, and I think that makes a lot of sense and also has been empirically shown, is when you think about differences differences not only of species, because that's not the point. The point is practice. Monkeys are practiced for many, many sessions. You can do that with humans, and then you start to see that humans also start to show this collapsing bound behavior. So practice is a very important factor to take into account in decision problems. And it will alter really the mechanisms, how these decisions are made. Another important feature we found out why this happens is for example, the speed with which you present your problems. So for example, imagine you make a choice between a house or a Ferrari, yeah? And then the next choice is, and you ask, okay, overall you should decide of all the items, pile A or B, which one you choose, right? Like you have multiple items that make it more and more preferable to go for pillar A or B. And you delay the information that is presented. And then most likely, when you delay this a lot, like Ferrari, 5 seconds, a house, 5 seconds, people will get annoyed. And they say, I want Ferrari, I just go for B, right? Without actually having seen all the evidence. So this is also be modeled with a collapse bound and not a fixed threshold. A very different strategy, right, than awaiting all the information and basing your decision on everything that you can see. So one cannot say animals do urgency or animals collapse and humans don't. That is just simply not true. It's really exciting discussion, and it's empirically to be shown what they actually do. And this is exactly what we set out in this last experiment. We wanted to test whether brain regions are associated with a fixed bound setting in this RLERM, in this reinforcement learning discrimination task, or whether they are actually collapsing their bounds, and which regions are associated with that. So for that, there are advantages. I'm just going through this very quickly, but just coming to the results now, because our goal, as I said, was to develop, first of all, a quantitative model that can capture reinforcement learning aspects and discrimination behavior, and also where we can estimate reward prediction errors. And we wanted to do this in this overarching group level distribution, so using Bayesian hierarchical modeling to bring this all in one framework. These are the model fits. If you're not used to looking at this is basically a quantile plot for the reaction time distributions for, first of all, the distributions for the percentage of accuracy, separately for speed and accuracy, for correct RTs and for erroneous RTs. And we see that the model prediction, the shaded area, is actually overlapping the empirical data very well. There's little uncertainty, which means that this model, at least for this type of implementation of the task, is quite suitable. And just as a last picture, or some of the last pictures, here are the brain results. So here are the really truly brain behavior relationships between these deep brain structures that I showed you that we mapped out in detail in the beginning. And these are not all 63, but these are, I think, 12 different structures for left and right hemisphere. And what I'd like to focus you on is just these red rectangulars. Now, what we see is that these different ROAs, regions of interest, so brain regions, significantly co-vary with a change in collapsing bound. And you may not find this very exciting. It looks a bit dry, I agree. It's not a fleshy brain blob. But I thought it was really striking because my previous research, so studies that I did before and could replicate, showed that there is no urgency term needed. But in fact, I could show that just a striatum correlated positively with a change in response thresholds, caution, meaning that this region, particularly on the deep brain, is preparing you for very quick responses. But now it turns out this is not the case, but in fact for these types of experiments you need to have this urgency term, and this urgency term is eventually the one that's driving these activations in these many different subcortical regions. And it's not even, it's one region, it's across the board. It's basically really the brain is on fire, if you want, down there. And that again attests the first part of my talk, that really more work is needed, that we map these out, that we better understand, that we stop globology, that we really better understand the plethora of activations, of co-activations that's happening. Another result, or just out of interest, so I told you about the thalamus, right? And all of you knew it. Now here's the thalamus, the way you can segment it. It's just one way to do it. And it's, again, people think there are 80 substructures. I'm only mapping 14, and I can't even get further because they get so small, it's impossible. But also here, and I could not find this in my previous studies, I think the sensitivity of this joint modeling really now gives insight that also the thalamus is heavily involved in these urgency terms, so in these strategic adjustments during decision making and across the board really relating positively to collapsing bounds, not fixed thresholds. And the same holds also for the so-called striatum. Again, I won't bother you. It's, again, a very big structure deep down there that I found previously. But again, I could not replicate my previous results, but I see, again, a positive correlation with these urgency terms. So this calls really a bit for a revision of thinking that these fixed thresholds are important, and calls also into question, how this has been imaged, including my own studies, including my own experimentation in the past, and that eventually more sensitivity is really useful to overcome also biases in my own work. And with that, I like to wrap up. I presented a bit about joint modeling and why I think it could be an exciting new framework. Again, it also doesn't have to be brain data, imaging data, expensive stuff. It can be coming from all sorts of different peripheral measures. I really am. I'm a bit an advocate of ultra high field. I'm excited because of the resolution, because of what can be done in terms of learning brain function. But it is scarce. It's very expensive. It's not necessarily also needed for understanding, problems that you are working with. It is just a way that I started doing this. And then very concretely, really these last results that I showed you, they diverge from previous results, and I do think that happens because this approach eventually may be much more sensitive and reliable compared to our previous work. And here are just a few people that I definitely need to mention, because you've seen, it is highly interdisciplinary. So this is the neuroanatomy team led by Anneke Alkema, the associate professor. She cuts the brain. She did all the hard work here, long work of doing all of this and bringing that all together. Pilou also helped a great deal. Pilou Bazan, he's a senior scientist. And then various modelers and cognitive neuroscientists that worked on all of these different experiments and models and tests. And with that, I'd like to thank you very much for inviting me again, for being so friendly, for so welcoming, and I hope thank you. Thank you for this. Well.
I'll start off. Can you hear me?
Yeah.
Thank you very much for this very illuminating speech on diffusion models and evidence accumulation models. But at the end of the day, I haven't followed this literature very much, but at the end of the day, there's still these issues as to what evidence is being accumulated, right? So for me, it sounds very much like an as-if model. There is no link directly to, let's say, if you are solving a navigation task and you have to go right or left, what's going on in hippocampus, for instance, where we actually know something. How much is known about this? Are people working in that direction as well?
Not that I am aware of. It's a very interesting question, and of course very relevant. And you are right? So spatial navigation, right? And of course, I mean, my first intuitive answer to that would be that exactly this last part that I showed, where you start to integrate, where you start to think about dependencies between trials, right, or observations. This is the way forward, because also for spatial navigation, right, you come from somewhere and you go somewhere, and there needs to be a dependency. Yeah, you are not point by point resetting, and then you are in a new decision situation, right? You eventually have some prior information how you should navigate through this labyrinth, if you're thinking about something like that. And I see exciting avenues exactly to first of all include the history of decisions, which is needed, I think, in order to solve this, and then the prediction for an upcoming decision that is informed of this history. And of course, there are ideas. There are ideas more in the memory literature that use different types of models. They are eventually also more neurally inspired, right, with spatial grid cells and things like that. I'm not an expert on this, so I'm very careful. But I think what is not taken into account is, so you can, of course, everyone has a model that's their own toothbrush, and eventually I heard someone saying a model like, you don't share this. Like, you love your toothbrush, you use it yourself, and that's it, right? But it is, of course, very nice if we're not having all toothbrush. I mean, it's nice if everyone has a toothbrush, sure, I hope so. But, you know, if we have models that actually are generalizable. And I have not seen sequential sampling models being used in that context, but I don't see why this can't be done. What it would require is, for example, to have a speed aspect to your decision. Because I think what mostly has been done is more looking at the quality of the decision, so, or the prior probability, calculating that, right? How probable is it that you go left or right? How probable is it based on a neural signal that informs making this prediction? And there is work, for sure, I know that, but not in such a behavior modeling framework, as far as I know, not with evidence accumulation models. Yeah, thank you.
Yeah, so I have a question that I hope it's not too stupid. So, so in this experiment, it's very clear cut that, okay, now you have a decision to make, and now you're presented evidence, but in daily life, in many situations, that it's not really clear that you have to make a decision. For example, what I'm going to have my dinner. Obviously, it's a decision that I have to make, but it's not on my mind. But on the other hand, I'm already accumulating evidence of what I may want for what I eat tonight. So, what actually fires the gun for? Okay, now that we have the threshold, and it now starts collapsing, and now the noisy diffusion starts.
If I understand you correctly, I think you're asking about prospective planning, right, and how that could be eventually be incorporated. So your plan, so the plan of something that is much later than in front of you.
Yes. So when do you think that believe that is a decision for you? It's a decision time.
Yeah, excellent question. There are, for example, studies. Well, old again, it's relative, but it is a study, I think, about 20 years ago. It's a Saccard, it was a Saccard task, beautiful work from Rangel's group, an economic decision maker, I think, as far as I know. And he had an experiment where he let people freely view at 2 different options. I don't know if it exactly relates to your question, but it is a bit like I'm looking at an apple, I'm looking at a chocolate, and they were asked, at some point you make a decision, and I'm just interested in how long you look at stuff, right? Which could be also dinner, right? You kind of, it's on your, at some point you know you have to eat. Let's assume you are at that point, and you are in the position you actually need to decide, what type of food or what do I pick from the menu? And so that's what he was interested in. It's not like just wants a card and that's the decision and that's what I'm going for, but he was really actually letting people view around. And the question was, does the last card, the duration, the fixation duration, does that predict the outcome of the decision? I think that's more natural than what I'm doing here, right? So that was a really nice experiment, I think, in this direction of accommodating a bit more naturalistically how you make decision in everyday life, yeah? Because you don't sit there and then, right? It's like you're weighing the options, the alternatives. And I think ISA cards are very powerful in that respect because you get traction of what your gaze is at. Other than, I wouldn't know, maybe there are models that also accommodate more this internal rumination process, right? That you actually think in this direction for a while and then you do something else. There was also a really nice experiment with a joystick, and they tracked basically the motion of where people put the joystick, right? There are multiple options where they basically could go. And so the paper was called Change of Mind, I think quite prominent in science. And they saw that people not perform like I showed you, right? This is highly constrained, I agree with you. But what they could show is exactly people go in one direction, they think, Italian might be nice, but then I had this yesterday, change of mind, going somewhere else. So they had, that empirical traction by looking at these traces of the joystick, how people navigated this to the end point. But still, this was implemented in a variant of evidence accumulation models. But I agree with you. Everything that I explained to you, or what I just explained, is experimental. Yeah, I would not like to argue that this is real life at this point. It's really.
I just want to ask whether this ISA card or other evidence, does this corroborate with the neuroscience of the diffusion process? Okay, now we can also see the neuroactivities for the diffusion.
That's also a very good question and a complicated one. So initially, 20 years ago, I would say yes, because what Michael Chadlin did, very prominent work also, he recorded from non-human primates from a region here. It's called interparietal sulcus or parietal cortex. Doesn't matter, but it's somewhere here. So he put an electrode in the monkey skull into the cortex, and he recorded from them while they did exactly these types of tasks that I presented, or these perceptual discrimination tasks. What he could show is that the neurons within this area showed basically a noisy accumulation process as the diffusion model assumes, and the behavioral data is modeled with. So one-to-one, right, seems like, the neurons do exactly what this theoretical model does, right? And then a lot of work afterwards was trying also to connect the dots tighter, a bit like what I'm trying to do here. So create models also where you use these spike trains or spike rates and you inform the behavioral models from these spike trains. Beautiful work, also interesting. But then it turned out that there is just not this homunculus. What is actually happening is that eventually we need to think in terms of brain networks, because others, for example, started to record from other areas in cortex. For example, the frontal eye fields that recorded from lateral prefrontal cortex. They also started to record from deep brain structures that I'm interested in, and what we see is a very similar signature in terms of noisy evidence accumulation. Now, having said that, you can say, well, that's great. Well, then it's just the entire brain, or like many networks, they do this, but the signature is exactly that. but also there has been a discussion whether this is now really a stochastic process, whether it's a step function, how to best accommodate, how to best model this. It's a field full of discussions, which tells me also that people are interested, but that there are many unknowns. And so what I'm presenting to you here is also not in any way an argument that I'm trying to equate the models I use to be biologically informed. There are no neurons that show this ballistic thing, or maybe a very specific type, but these are not the ones that I'm looking at. This is an assumption, right? This is a mathematical assumption in the end also that is made, and I don't think the brain works like what I present here. Yeah, just to be very clear. I wish I would know, but I don't. But it is, again, still a way of getting closer to the real mechanism and the sub-process of decision-making that is implemented in the brain. Because else I'm just standing there and I have this one reaction time and I don't even know what happened there.
Okay, thank you.
You're welcome.
Thank you very much for the talk. I really found it interesting. I am more kind of a applied psychologist, so I'm fascinated, and I always wanted to know what goes on in the brain regarding my models.
Me too.
Exactly. So you said even mapping the brain like you do it will take 3 lifetimes. So will I ever have the chance, or even like my grandchildren, to map a decision process in the brain?
You have hard questions. I like it. I wish I had excellent answers, but I don't. It's an iterative process, right? And it's a lame answer, but it is what it is. And that is the hard way I learned exactly with these lifetimes, right? And it was an eye-opener, really, to come from cognitive neuroscience and to think, oh, wow, we have now this non-invasive, beautiful way of mapping the brain. That will change the world. To be honest, many people are more and more questioning whether this ever can be accomplished or if it makes any sense. But then again, I mean, there are perfect scenarios that I can imagine that really would resolve some questions that I also have, right? Like, for example, how does it really work? But it would be highly invasive, so it's a no-go. And not necessarily even would it really answer to the fullest how exactly, where exactly, in what interactions, right? Because one thing is clear, the brain is highly interconnected, yeah? But if you don't know where the seeds are, if you don't even know, where to start from and what to connect with what, where are we then, right? So, I think the best and most humble answer I can give is an iterative process, and I think it's also then a process in that sense that you dare to dream, but that you also have the courage to basically just accept that while you see somewhat of the picture, you study a very specific, tiny aspect of this. And there will not be the point, I don't think, in my lifetime that I really go to bed and think, oh, yes, I know how it goes.
Thank you.
I know how it is. I wish. It reminds me of conversations with my mother. When I went home, she's like, "Yeah, what did you find out?" And like, she wasn't a scientist, it's like, let's talk about Parkinson. I think that's very relevant. It's a beautiful question, but that's the most honest answer I think I can.
Thank you very much.
Yeah.
So you said that you don't really believe that this kind of racing models reproduce the neural data, but I actually want to ask the opposite. So there was an earlier question about applying this to subjective preferential decisions, which economists would be much more interested in. And I don't know whether you remember the old mathematical psychology literature, but already in 1992, and I'm showing my age now, Marley and Colonius had a very, very destructive theorem about those models, showing that if you do that, which is called a random utility model, and add racing models in order to generate the response times, anything goes. You give me any data, does not matter what it is, I can cook up a model which will generate exactly that. Which for economists and mathematical psychology is very bad news, because it means that those models have zero biting power. So my question is the opposite. What can you tell us from neural regularities which should be fulfilled so that you can refine those models and increase their biting power?
Yeah. Excellent, and this is also really a reoccurring question, right, for the neuroscientists. Like, what do we learn from this, right? Like, why do you need this? What is the use? If I, for example, have such powerful models, can I actually constrain them, right? Can I use this modality to actually help? To help this side as well. So, can I go back? No, I can't go back. Can I go back? Can I go back in my slides, please? Oh, yeah, da.
Oh, not working.
Nee. Yeah. Oh, yeah, perfect. Thank you. Yeah. I'm trying to come up with a most concrete answer, and it's just one out of many that I could think of as a potential avenue. And there's only 1 or 2 papers that I know that tried that, and they were not necessarily very convincing, but I haven't given up on this, because I believe that that exactly this could be a fruitful avenue. And that is that we're looking actually at the neural signal for a moment. This is exactly what you see here. I skipped the slide, but this is how I model my time series of my brain data. Basically, I'm using, in the simplest version, the general linear model. And that has specific parameters again. And it basically, the parameters signify, for example, the amplitude, the peak, so how much is it active. I have a parameter that gives me an overview of the dispersion, so the width of the curve, because basically what we want to estimate here is the measured signal on a particular event, for example, Q presentation multiplied by the beta weight and some error term. Now, when I model this data, I use a function. I use a hemodynamic response function. And this response function, you can now come in and speculate. Look, if I give you the speed accuracy manipulation, right, and I think this works the way it works in the DDM, but I now take, for example, I speculate, well, the areas really should change in amplitude. So I take the amplitude parameter of my GLM and I inject this, and I hypothesize that, for example, the boundary separation should be affected by exactly only this parameter and nothing else, right? And then also I can compare it across different models, also the ones maybe you mentioned, right? And can selectively show what the influence of my neural model parameter is on a model parameter in the behavior. And with that, I can create very concrete predictions how a particular change in the HRF, which I actually know. I know what the change there, what parameter change actually does onto my signal. Certainly the amplitude. Then I can at least, to some extent, selectively showcase, okay, that, has the effect, or selective influence on changing the response parameter or urgency parameter that I expected to do. It's still inferential. I'm not sure it really actually does answer truly your question, because we're still looking to some extent at correlations here, right? You can, but it is trying to come more and more to concrete hypothesis how selectively the neural signature, and I'm talking bold here, right? You can also, of course, do this with EFIS and whatnot, selectively influences exactly the prediction of the behavior. I think that could be really powerful. And maybe finally a bit informative, more from this black box what we have up here, and make use of neurally informed models. without over-parameterizing them, because I'm very, I'm not knowledgeable enough, but I'm just really scared of models that, you know, expand in a model space that I eventually really can't selectively affect anymore, and where I exactly, you know, just hocus pocus roll out, you know, the prediction that I want, and I don't understand really under the hood experimentally also what happened here, yeah, because so many possibilities are given. But I think that could be a beautiful way of constraining these realms more. Whether it would be the distinguishing factor to differentiate, for example, between the models that you mentioned, right, and the ones that I discussed here. Yeah, maybe also really an empirical test. But I do think that the neural side could have enough information as of yet as to try this. Yeah, we're not tapping in the dark here anymore. So I see some possibilities there.
Thank you. Any other questions? If not, thank you very much, Birte Forstmann, for an excellent introduction to the conference.
So, I have a couple of things to tell you. First of all, I want to say thank you to you and give you something.
Thank you so much.
And this for you.
Thank you.
And also that present.
Oh, that is beautiful.
Yes.
Castellón?
Yeah, it's here with.
Oh, beautiful.
Okay.
Thank you. Thank you very much. Thank you. Nice. Thank you.
And then a couple of words, because since we started a bit ahead, party is not ready yet, so I'm going to tell you a couple of things. The first thing that I would like to tell you is that you have here probably some codes. Those are to be connected to the free Wi-Fi of the university. You have to pick the network called UHI, like the university. And then the first set of, the first word is the user, and the second one, the password, okay? And with that, you have free Wi-Fi all the time. And the other thing I would like to inform you is that on Thursday, we have a gala dinner in the middle of nowhere. And even though you bring your own car, it's sort of mandatory to take the transfer we offer, because it's in the middle of nowhere, in very dangerous road. So don't be overconfident, please pick the bus we offer you, okay? And the last thing I have to say is that if we go out and party is not ready, I ask you to be a bit patient, because they are working, they're doing their best to have it finished as soon as possible. Thank you.